Hello pharma aspirants, welcome to online GDC platform. So as a series of Pharma Thunderstorm, we are going to discuss some of the key concepts in the subjects of your pharmacy course. Okay, so as part of that, today in our pharmaceutical analysis subject, we are going to have a discussion on the topic tolerance limits for the glassware calibration. Okay, so as a Pharma Thunderstorm series, we have included this topic in this, and in this series you are going to get a key idea as well as quick tricks as well as some of the key concepts also you are going to learn, so that all these concepts will be helpful for you in cracking your competitive exams like MRB, RRB, as well as GPAT, 𝙂𝙋𝙬𝙏, Pharmacist, okay, AIMS Pharmacist, all these professional courses whenever you are going to attempt. So let us start our today's session in our Pharma Thunderstorm series, the tolerance limits for the glassware calibration. Okay, generally in our laboratory glassware, whenever we are going to use, there are n number of the glassware are there. Okay, like starting from the reagent bottles as well as normal sample containers, okay, and so many equipments are also there, but we are talking only about the glassware, means the glassware which are made up of the either type 1 or type 2 glass. Okay, so only those two are allowed in the laboratory experiments. So in that you are going to see two types. This type 1 glassware is only the generally preferred. In this also you are going to see two types of the glassware, that is, those are nothing but class A and class B. Okay, so class A and class B type of glasswares means these two are allowed in the laboratory experiments. In this class A glassware, which are majorly made up of borosilicate type glass. Okay, borosilicate glass means it is highly resistant towards the chemicals whatever we are going to use in the pharmaceutical laboratory, as well as it is also thermally highly unstable also, because under certain condition thermal expansion, it is also going to be one of the major problem for the glassware. So this thermal expansion coefficient, so in for this borosilicate glass, whenever we are talking about the problems, okay, so the borosilicate glass, the thermal expansion coefficient, it is going to be in the required level. Okay, so that's why this is the highly preferred glass for the manufacture of all the laboratory glasswares. And class B glassware, it is also there. This is actually made up of soda lime, and these glasses are also going to be used in our laboratory work, but there is a clause for it. In order to use the class A or class B glassware, we should ensure that these glassware are properly calibrated. Okay, so after calibration only you have to use these n number of the glassware whatever we are going to use, like measuring cylinders, volumetric flasks, as well as burettes, okay, pipettes, 1 mark pipettes or graduated pipettes you are going to use. So all these types of the glassware should be properly calibrated, then only you have to use it for the laboratory experiment, because in the laboratory experiments we are going to deal with the samples or solutions which are going to be used for the human consumption. Okay, for that purpose all these accurate measurements are required, because whatever the pharmaceuticals we are going to manufacture, all those are the dose dependents. Not only in the analysis department, in the manufacturing department also they are going to use some of the glassware. All the glassware whatever they are going to use, which are going to be properly, that means accurate measurements are required, means they need to be calibrated. Okay, so class A whatever the glassware we are going to use, majorly these are class A glassware are provided with a calibration certificate of analysis. They are going to provide a calibration certificate. The manufacturer, whoever is providing you the glassware, will give you already the COA, certificate of analysis. Okay, calibrated certificates are available for the class A glassware, whereas for the class B which are made up of soda lime glass, these are have to be calibrated by us. So for that we should know about the procedure of the calibration and what are the tolerance limits. Okay, and not only for the class B, you should be aware of the tolerance limits for the class A also, but already in the COA they are providing you the tolerance limits. So let us see the what are the different that means in order to proceed for the calibration, what we require, that means whenever we are in the different climatic zones, okay, zone 1, zone 2, zone 3, zone 4, the world has been entirely divided into the 4 zones. Okay, so we are in the Indian side, means we are at the temperate region, high humidity we are going to observe at our conditions. Okay, so according to our Indian climate, that means we are going to use the glasses whatever in the laboratory, those also should be chemically resistant, and the temperature is also going to affect the expansion of the glasses. Okay, so for that purpose, according to our standards, that is nothing but the Bureau of Indian Standards, BIS, Bureau of Indian Standards, they have concluded that in their document, all the volumetric apparatus, whatever we are going to use, all the volumetric apparatus are normally calibrated at the 27 degrees centigrade. So if you observe the glassware, whatever you procure from the manufacturer, on the glassware itself you can see that there is a borosilicate glass class A, they are going to mention at 20 degrees what is the exact volume. They are going to mention the capacity of the glass also. Okay, that means if you are going to observe the image of the particular glassware like this, they are going to give what is the grade according to standards. They are going to maintain ISO 1042. They have just mentioned here, you can observe here. Okay, this is a class A, like class A grade glassware this is, and 20 degrees centigrade they are going to use, and this is made up of the borosilicate. Okay, and what is the capacity of that, whether it is 250 ml, 100 ml, the capacity of that also they are going to mention, as well as who is the manufacturer of the glassware also they are going to imprint on the particular this glassware. Okay, so if you see the all the details on the glassware itself, you will come to know that whether it is a class A or class B, and what is the capacity of that, what is the tolerance, or also sometimes they are going to give plus or minus. Okay, not for the all the glassware, this is actually high 250 ml capacity, so they have given the plus or minus some. Actually it is not clearly visible, but they have given the tolerance limits also. Okay, but at 20 degrees they have performed the tolerance limits, and they have provided you the limits. And according to our requirements, okay, that means according to Bureau of the Indian Standards, we need to perform the calibration. That means all the volumetric flasks or volumetric apparatus we are going to use in our laboratory experiments, they have to be normally calibrated at the 27 degrees centigrade. Okay, 20 degrees centigrade means that is not suitable for our climatic zones. So pharmacopoeial assays, generally whenever we follow the pharmacopoeia, mostly for our compounds, means Indian pharmacopoeial compounds, whenever you are going to see the monographs or whatever those are. So in the monographs you are going to find different n number of the tests also. So wherever the pharmacopoeial assays are involving the volumetric measurements, okay, so those volumetric measurements require the uses of accurately calibrated glassware. Accurately calibrated glassware only you have to use. They have mentioned use 250 ml volumetric flask. So what it means you need to use a properly calibrated glassware only you have to use it for the particular assay. Okay, so pharmacopoeia suggests that after proper calibration and the different procedures are also they are going to give you that. and in that they have mentioned about the tolerance as well as reference temperature also, because this glass, it is going to majorly affected by the temperature. So we need to majorly worry about this temperature also. So in order to know about how the temperature it is going to affect, and it may affect the volume of the solutions at any condition, at any stage also. Okay, so that's why you should know about the what is the working temperature, right, and what is the suitable experimental condition where you have to perform the particular analysis. Okay, so in order to carry out all these things, you should aware of all the basic things in this glassware calibration. So let us know first what is called reference temperature. That means the temperature at which the flask for the or whatever the flask or glassware apparatus it is there. Okay, so the temperature at which the glassware is intended to contain or deliver a volume that is equivalent to its nominal capacity. Okay, that means the temperature, what is called the reference temperature, the temperature at which the glassware, whether it is a flask or measuring cylinder or a pipette or whatever it is intended to contain or it is intended to deliver a volume, that is a volume which is equivalent to its nominal capacity. If our nominal capacity of the flask is 10 ml, okay, so the temperature at which the reference temperature should be given in such a way that at that particular temperature exactly at it is delivering the 10 ml of its volume. Okay, so that is called the reference temperature. So most of the borosilicate glass you are going to see the temperatures at 20 degrees centigrade with plus or minus tolerance limits they are giving, right. So let us see what is the tolerance limits. So these tolerances actually nothing but maximum permissible errors. Okay, that means whenever we have already measured some of the volume after calibration, then from the nominal, whatever the nominal capacity it is there, if you subtract from the observed value, that is called as the tolerance limit, maximum permissible error. It could be in the plus or minus values. Okay, either negative side or positive side. Let us see what is called tolerance. Actually, the difference, this is actually the difference between the actual volume, that means the capacity which is intended at the reference temperature, the actual volume, and the indicated volume. Okay, that means whatever we have indicated means the nominal capacity. Okay, what is the nominal capacity, that is indicated by the manufacturer, and the actual volume means what we calculated actually at the bench top. Okay, so the difference between the actual volume and the indicated, who have given the glassware, particularly the manufacturer, the difference it is going to be taken at the prescribed reference temperature. Okay, so at the prescribed reference temperature, if you are able to calculate the difference between the actual volume as well as the indicated volume, that is called as a tolerance. The tolerance, it is also called as maximum permissible error. It could be either plus value or minus value. Okay, either towards the positive side or negative side also you are going to observe these tolerances. So these tolerances for the glassware calibrations are very, very important. If these are within this tolerance limits only, then you are allowed to use the particular glassware. Okay, for that purpose actually so many standards we are going to follow according to if you are in which place you are working, that is according to that you have to follow. Okay, so if you are in UK, US or Japan, so according to their guidelines only you need to follow the tolerance limits. Okay, so majorly National Institute of Standards and Technology, so that means everyone want to adopt the highly recommended standards, right, so that in future or whatever the drug development departments are there, all these whatever the research we are going to carry out, these won't have any problem in the people whoever working out of or near to our places also, different zones. Okay, majorly actually if you are manufacturing a product at your place, you want to sell the product at all the zones also. Okay, so your product should be acceptable everywhere. So in such a way that you need to maintain your standards. So according to that, if you acquire or if you adopt to all the standards, strict standards if you follow, then everywhere the method of analysis or method of manufacturing, whatever it is there, it will be acceptable. Okay, so National Institute of the Standards and Technology, it is going to be called as NIST. Okay, it has given some document which is nothing but selected procedures for the volumetric calibrations. How you go for the calibrations of this volumetric glassware. Okay, so that is actually the document number NIST IR 7383 in 2019. So earlier also they have given so many reference documents, but after recently, these are the recent documents we need to follow. And one more standard agency, that is ASTM International, now it is called as, but formerly it is known as American Society for the Testing and the Materials. Okay, ASTM standards for all the standard weights and measurements, also metrology departments are also, they are going to follow these ASTM standards only. So ASTM standards, you are going to find so many documents are also related to this calibration of the glassware. So if you find in 2017, okay, the document number is 𝙂𝙂𝙎𝙬. So version 10. So standard specification for the laboratory glass volumetric flask. So specifically this is document for the volumetric flask, and this one more document 2018 they have given for the specification for the glasses in the laboratory apparatus. Okay, so like that they have subdivided for different categories, like E54201 in 2021 they have given the document, that is standard practice for the gravimetric calibration of the laboratory volumetric instruments. That means gravimetrically how we can proceed for the glassware calibration. Okay, so that is a document. And one more document, that is E694 18th version, standard specification for the laboratory glassware volumetric apparatus. Okay, so all the volumetric glassware they have included here. So like this different types of the documents are also there, different standards we need to follow. And one more, that is standard specification for the laboratory glass graduated cylinders you are going to see, measuring cylinders. Okay, so graduation cylinders you are going to observe for this in 2019, in 2017, 18, 19 totally actually they have subdivided, and in all this 2021 they have completely gravimetric calibration of the all the laboratory volumetric instruments. So the concise document they have released. Okay, so these are the reference documents you can follow while adopting the procedure for the glassware calibration. Okay, whatever the procedure you follow, but you need to make sure that it is according to the tolerance limits. Okay, within the specification limits it is there, otherwise you are not supposed to use the particular glassware. Okay, so coming to our Indian standards. Okay, BIS, Bureau of the Indian Standards, if it is there, actually it is a statutory body which is established under this Bureau of Indian Standards Act 1986. Okay, which is promoting majorly the harmonious development of all the activities of the standardizations, related to the standardization, so that quality of the product it is going to be used by the people. Okay, so all the certifications are also going to be done for all the goods and attending to the connected matters, whatever it is, not only the glassware, they are going to maintain this hallmark in the gold also, like that they are going to follow in so many areas. Okay, so one more standard document it is also there, ISO, internationally accepted document, ISO, International Standards for International Organization for the Standards. Okay, so 4787 in 1984 actually they have given laboratory glassware one document. In this they are going to that means all the procedures related to how to calibrate the all the volumetric glassware, methods for the use and testing of the capacity, they are given in this document. And the same document we have adopted, that means that has been adopted by Bureau of the Indian Standards also, so that we are also following the same international standards. Okay, so that we are meeting up to the all the international standards there. And if you want to, that means if you follow all these standardization limits, okay, so that you can make sure that whatever the procedures you are going to follow or whatever the procedures already established in your laboratory, these are meeting the requirements of internationally accepted limits, okay. And all the standards will help us in giving the high quality products, okay. So that is the main motto of this. In order to go for the calibration of the glassware or whatever the instrumental calibrations, whatever we are going to follow, but all these are going to be helpful in giving the high quality products only, okay. So tolerance limits, if you are taking into consideration, means according to Indian Pharmacopoeia, what are the tolerance limits? Let us see in our today's session. So these are the some of the tolerance limits for the volumetric flask, okay, tolerance as per the IP, Indian Pharmacopoeia. So volumetric flask, if you are talking about, means these are different capacities are available, like 5 ml, 10 ml, 20 ml, 25, 50, 100, 250, 500 and 1000 ml, okay. So 2000 ml it is also available, but okay, that is not preferred one, okay. And actually anywhere glassware which is having high capacity as well as low capacity, those are not preferable, okay. So within the range of the capacity, that means 5 to 500, that is the optimal range in the normal laboratory experiments, okay. But very high capacity glassware like 2 liters, 5 liters if you are going to use, means in that range of the tolerance, that means whatever the tolerance limits are there, those are also going to be more, that means the window whatever it is there, that is also going to be more will be there, so that errors could be very less, okay. And because pharmaceutical analysis, whenever you are going to do, means we are going to perform at the mgs levels, micrograms levels. So that's why what are the optimum ranges for the laboratory glassware, that we are going to discuss, okay. So if you want to see the 5 ml capacity volumetric flask, we know that it is going to be like this, a flat, and it is going to be, so in this volumetric flask you are going to see the mark here, okay. So one lid is also available for the volumetric flask. So always try to weigh the content in the volumetric flask. Along with that, if you want to weigh the volumetric flask, means try to put the lid also. So along with the lid only you are supposed to weigh it, okay. So volumetric flask tolerance limits, if you want to see, that is class A and class B, both grades are available. So if you find this class A for 5 ml, the tolerance limits is plus or minus 0.025 ml, okay. So and 10 ml also the same 0.025 ml you are going to see, and class B also if you see same 0.05 ml for both the 5 ml and 10 ml, whereas for the 20 and 25 ml if you see, these are also going to be the same thing, okay, 0.04 for class A and 0.08 for the class B. And 50 and 100 if you see, these are going to be somehow different, okay. 50 if you see the 0.06 plus or minus 0.06 ml. So somehow little bit here you can see 20 ml 0.04, 25 ml 0.04, for 50 ml 0.06 you are going to see, and for 100 ml if you see 0.1, whereas 250 ml 0.15, whereas for the class B grade you are going to observe means 0.2 ml. So almost it is doubled. Here also for 50 ml it is doubled, 25 ml also here it is double the case, and 10 ml also the tolerance limit is almost double. So comparatively class A and class B if you observe, the tolerance limits are double than the one than class A, okay. So 0.1, this is 0.2, 0.15, 0.3, 0.25, 0.5, 0.4, 0.8. So in this way you can remember very easily. So once already in the some of the competitive exams they have asked the tolerance limits for the some of the class B volumetric flask at 200 ml volumetric flask, what is the according to Bureau of the Indian Standards, what is the tolerance limit. Okay. So, in such a way you are going to face the questions in the competitive exams also. So, this session is going to be helpful in one or the other way. Okay. So, this is about the tolerance limits for the volumetric flask, and okay. So, let us see how this tolerance it is going to be calculated. Till now we have discussed that okay, tolerance within the tolerance limits means the glassware is allowed to use it, but how we are going to do the calibration actually. So, whenever you are going to ask to do the calibration of any type of the glassware, the minimum requirement you need to take, that is a calibrated weighing balance. Okay. So, a calibrated weighing balance first of all you should have, and automatically you need to use the distilled water. Okay. So, distilled water you have to take it, and what is the density of the water at the particular temperatures, that is also you should aware of this. So, but always 27 degrees in the laboratory it is difficult to maintain. So, we should have a table of the density chart of the water. So, in that you should have the information related to at which temperature, that means at working temperature. If your working temperature is 25 degrees, then what is the density of the water you should know. If the working temperature is 22 degrees or 24 degrees or 28 degrees, so what is the density of the water at particular temperature, you should note that from the chart. Okay. And you should have the calibrated weighing balance and distilled water, as well as you need to calculation part also it is required. For that purpose it is required, go for the calculator also. Okay. So, otherwise in all your computers also it is being provided, and once you have these calibrated weighing balance, distilled water, and whatever the glassware you want to calibrate, then by using a differential weighing method you are going to calibrate the glassware. So, for that what you have to do, that is first of all weigh the empty glassware. Suppose let us say if you have taken the volumetric glassware, volumetric flask. Okay. So, volumetric flask along with the lid we have taken. So, let us say 25 ml volumetric flask we have taken. Okay. First of all take the weight of empty volumetric flask, that is going to be called it as W1. Okay. So, then fill the flask up to the mark, up to the given mark, fill the flask with the water, plus water you have to add up to the mark, up to the lower meniscus, because water is a colorless solution. Okay. So, then again measure the weight, that is going to be considered as a W2. Okay. So, then you have to consider what is the net weight of the water, W2 minus W1. If you subtract the W2 from these two, W2 and W1, you will get the actual weight of the water. But actual weight of the water, that is according to the temperature. Water is having different densities according to the different temperatures. Okay. So, W2 minus W1 divided by density of the water at the particular temperature, at the whatever the working temperature it is there, at that particular temperature you have to take the density of the water. Okay. So, if you divide the actual weight of the water divided by density of the water, then you are going to see the exact weight. Okay. So, the exact, not the weight, volume, the exact volume for this volumetric flask. So, in this way we have to proceed for the calibration of any type of the glassware. Okay. So, not only volumetric, burette also you are going to take in the same way. First take the empty weight of the burette, then fill it with the required water up to the mark, then again weigh it. So, differential weighing you need to take it, thereby divide by the density of the water, you will get the exact, that is called as actually actual volume. Okay. That means the calculated value, calculated capacity it is. Okay. So, this is the actual volume we will get right now. Then whatever the theoretical value, that means the nominal capacity volume. What is the nominal capacity? So, for that if this is actually the 25 ml. So, how much we got? Let us say we got 25.0001. Okay. So the nominal capacity is 25.000. So you need to subtract this, then how much you are getting? Plus 0.0001. Okay. So whether this is within the tolerance limit or not. So in this 25 ml volumetric flask, if you observe, our tolerance limit is plus or minus 0.04 ml. Okay. So this 0.0001 is less than that of the 0.04. Plus or minus 0.04 means what? Minus 0.042 plus 0.04. Okay. So up to that, that is the range. So this 0.0001 means this is within this limit. So in that way, you need to proceed for the glassware calibration, and these are the tolerance limits, how you are going to check. Tolerance limits, specification limits, whatever it is, we are going to call it as, but for the, especially for the glassware calibration means this is called the tolerance limit. Okay. So within the tolerance limit or not, we need to check it out. Whether if it is within the limit means you have to proceed further for the usage of the particular glassware. Right. So I think this slide is given the full-fledged information for you. Now let us see the tolerance limits for the 1 mark pipettes. 1 mark pipettes as per the Indian Pharmacopoeia only. If you observe this slide, class A and class B, both grades they have given for the 1 mark pipette. Okay, 1 mark pipette means generally you are going to see the bulb pipettes you are going to observe. So whatever the bulb pipettes are there, these are going to have the 1 mark pipettes. Okay, so cone shape ending will be there. So these are going to have a provision. So 1 mark pipettes means a single division. 10 ml capacity bulb pipettes, these are going to be called as bulb pipettes. 5 ml capacity bulb pipettes, 1 ml capacity bulb pipette is also there. So according to IP, they have given 1 mark pipettes means bulb pipettes of 1 ml, 2 ml, 5, 10, 20, 25, 50, 100 ml capacity. Okay, generally we don't go for the this 50 and 100, up to 20 and 25 we are going to use the bulb pipettes. Okay. So let us see the tolerance limits for this class A and class B grade for this bulb pipettes. So if you observe the class A, highly 0.007 ml only, plus or minus, and if you observe the class B, almost double, 0.015. Similarly for 2 ml 0.01, and this is double. 5 ml 0.015, and the class B it is double 0.03, and 10 ml 0.02, and for class B it is double 0.04, 20 ml 0.03, and for class B it is 0.06. Okay. So almost all the values for the class A are there, and class B values are double than that of the class A. Okay. For 100 ml if you see 0.08, then 0.16 for the class B grade glassware you are going to observe. These are the for the 1 mark pipettes, that means bulb pipettes, 1 mark pipettes you are going to observe this, and we are going to see the next, we will see the next slide, so that the graduated pipettes, how the tolerance limits are going to be observed. Like graduated pipettes, you are going to see the subdivisions. Okay, subdivisions also going to be, sometimes you are going to observe the 0.01 ml subdivisions also will be there, and for few pipettes 0.02 ml subdivisions and 0.05 ml subdivisions are also some pipettes you are going to see. Okay, if you are going to go for the 25 ml graduated pipette means you are going to see the subdivisions of 0.2 ml. So the capacity increases, the graduation is also, interval is also going to be increased, and here class A and class B grade glassware, if you see the different tolerance limits for this graduated means for 1 ml it will be highly very narrow range you are going to observe. Even though for the bulb pipettes if you see, that is also going to be in the case of the 0.007 or something like that only we have observed. Yeah, here the same thing for bulb pipettes, here also 0.007, and for graduated pipettes also there is a 007, but class B there that was the double thing, but here 0.01, somehow it is less than that. And for 2 ml if you observe 0.01 and 0.02, here also if you see the 2 ml the same limits we have seen here, and for 5 ml 0.03 and 0.05, and for here somehow a little bit more strict limits we are going to observe for the 1 mark pipettes. Okay, rather than the graduated pipettes. For 25 ml, the tolerance limit is 0.1 ml and 0.2 ml, and for bulk pipettes, if you observe, these are only 0.03 and 0.06. Okay, very less range of the tolerance they have given. So in this case, the 1 mark pipette and graduated pipettes have the tolerance limits. And finally, if you observe the burettes, okay, so pipettes, burettes and volumetric flask, these are the highly used glassware in the normal analytical division. Okay, so burettes, whenever you are going to use the burettes for the volumetric titrations, generally we are going to perform. So these are the tolerance limits according to the Indian Pharmacopoeia, which are also having some divisions of 0.05, generally 0.05 for the low capacity, that is 10, 25 you are going to see, and for 50 and 100 the subdivisions will be either 0.1 and 0.2. Okay, so 10 ml burette if you take for class A 0.03, for class B 0.05, the same thing 25 ml also, both are same. Okay, as well as for 50 if you see 0.05 and it is almost double 0.1, and 100 ml 0.1 and double for the class B 0.2. Okay, so these are according to the Indian Pharmacopoeia limits, and this values actually according to the UK. That means if you are in the towards that UK side, then their own standards you need to follow. Okay, so they have given some of the tolerance limits for the class A and class B. They are going to use the, not the ml, they are going to use the centimeter cube, that is also 1 cc. We, in our terminology, we are going to call it as. Okay, so burette capacity, if you are in terms of only burette, I have listed out here. So 5 ml, 10 ml, 25, 50 and 100 they are going to use. So if you take the 5 ml burette, 0.02 and 0.04. Okay, so whereas for 10 ml if you take our limits according to Indian Pharmacopoeia 0.03 and 0.05, but their limits if you see 0.01 and 0.02, very narrow range you are going to observe. For 25 ml, but these two are same, 0.03 and 0.05, 50 ml 0.05 and 0.1, these are also similar, and 100 ml also same, 0.1 and 0.2, but at the lower volumes if you see, the accuracy is more here. Okay, so that's why if you follow the narrow range of the limits, that means more accurate results if you are able to perform, means your product is also going to be a highly qualified product. So that is the main motto of putting all the standards. Okay, so according to the different standards we need to follow this. For that purpose only they are going to give the tolerance limits. Hope this session is useful for you. Okay, so in our Pharma Thunderstorm series, we will meet with another interesting topic in the next session. Thank you students, thank you for attending.